Hello and welcome to another exciting Gen webinar. This one is entitled Combating SARS-CoV-2 Variants, Neutralization Testing from Alpha to Mu. This webinar was made possible through sponsorship from Integral Molecular. I'll be your host for today's event. I'm Jeff Bogaliskas, Technical Editor for Genetic Engineering and Biotechnology News, which is currently celebrating its 40th year, bringing you the latest information on advances within the life sciences industry. Now, while the various vaccines against SARS-CoV-2 have certainly had a significant impact on the slowing of the spread of the virus, the COVID-19 pandemic is far from over. And the continued development of advanced research tools, diagnostics, and therapeutics is critical to controlling and even potentially eradicating this disease. Tools that allow investigators to rapidly screen immunological mixtures for effectiveness against the SARS-CoV-2 virus and its numerous mutant strains. And today, our presenters are going to share with us their experience with some of these techniques that are helping to advance COVID-19 vaccines and therapeutic development endeavors. Let's meet our guests for today's event and find out more about who they are and the work that they do. Dr. Haley Crawford will lead things off today. Haley is a project leader in virology at Integral Molecular, where she manages the company's extensive Reporter Virus Particle, or RVP, portfolio. Haley spearheads all aspects of the SARS-CoV-2 Reporter Virus Particle product cycle, from product development through manufacturing and quality control for current and emerging strains. And today, she's going to tell us a lot more about RVPs and how they are being used to test neutralization titers of antibodies and vaccine sera. Haley will then hand things over to Dr. Pavel Nikitin, who is the Senior Director of Antibody Engineering at Immunome, where he heads the Antibody Engineering Group, currently developing a cocktail of therapeutic antibodies against COVID-19. Today, Pavel is going to give us some insight into how he and his colleagues are using memory B cells to develop antibody-based therapeutics. And then wrapping up the presentations will be Ufei Sheng, a research scientist in the Department of Cell Biology at the University of Pittsburgh School of Medicine. Ufei is a member of the Proteomics and Nanobody Bioengineering Lab of Dr. Yishi, where she and her colleagues identified a panel of ultra-potent nanobodies targeting the receptor binding domain of SARS-CoV-2 and showed the ability of the nanobodies to neutralize SARS-CoV-2 variants. Ufei is going to tell us more about this exciting work later on in the webinar. But first, before we get the webinar underway, I want to remind the audience to submit questions for our Q&A session, where all of our panelists will be on hand to try to answer as many of your questions as possible. So, if you have a question about any part of the presentations today, you can send them in at any time. You don't have to wait until the end. All you need to do is type your question into the Ask a Question box and then hit Submit. It's that simple. All right. Now that we have that all well in hand, let's turn the presentation over to Haley. Haley?
Good morning. I'm Haley Crawford from Integral Molecular, and I'm excited to tell you about the uses of our reporter virus particles for rapid and safe neutralization testing in the setting of the SARS-CoV-2 pandemic. Integral Molecular is a biotechnology company founded by virologists in Philadelphia. We have been producing virus-like particles for 2 decades and reporter virus particles for 11 years. Our RVP catalog is extensive and continues to grow. We are proud of the relationships we have built over the last 20 years. We have multiple academic and industry partners who trust us to work with them on their research. We publish papers on our own research and also collaborate on publications and patents with our partners. We have over 50 publications on viruses and over 250 publications on all of our technologies. Today I'm going to briefly tell you about our SARS-CoV-2 reporter virus particles, or RVPs, how they are used to measure neutralization, and our large and ever-expanding catalog of SARS-CoV-2 variant RVPs. I'll describe the FDA guidelines about the usage of RVPs for testing emerging SARS-CoV-2 variants, and I will then hand over to two of our customers to present their data. Our RVPs are fusion-competent pseudoviruses that are capable of one round of infection of target cells and are therefore safe in a BSL-2 environment. The HIV capsid, or core, contains a reporter gene that is expressed after membrane fusion, either green fluorescent protein or luciferase, and this enables detection of infected cells. The RVPs have been optimized to overexpress the membrane protein of interest on their surface, in this case, the SARS-CoV-2 spike protein. They contain no other genes from the virus of interest and cannot cause disease. The readouts of the infectivity and neutralization assays are given in relative light units when using luciferase RVPs detected via a luminometer in the left-hand figure, or as a percentage of infected cells when using GFP RVPs detected via flow or microscopy in the middle figure. We work in 384 well plates for high throughput assays, but many customers use 96 well plates. Our RVPs can be utilized in infectivity assays to study fusion and entry events, and in serum and antibody neutralization assays. We have VSV RVPs available as a negative control for neutralization assays. And with every first order of RVPs, we will send you a vial of our ACE2 HEC293 T cells. All of the optimized protocols are available on our website, and our scientists are also available to help you troubleshoot your assays. Our SARS-2 RVPs have been shown to be comparable to the gold standard plaque reduction neutralization test, or PRINT, by our customers at the University of Pittsburgh. Yufei Zhang will be presenting her data to you a bit later. PRINT requires live virus in BSL-3, it isn't high throughput, and it's laborious, while our RVPs are safe, high throughput, and easy to use. If your goal is to obtain reproducible antibody or serum neutralization titers, RVPs are optimal for this. We measure the NT50, or neutralizing titer of our in-house antibody, that neutralizes 50% of the RVPs. In the middle figure, you can see that we observed similar NT50 values for neutralization experiments performed on 5 independently derived lots of SARS-2 D614G variant RVPs. In the right figure, neutralization was performed using various volumes of SARS-2 RVPs from a single lot, between 5 and 40 microliters per well, and the curves and NT50 values were almost identical, showing the reproducibility and reliability of the RVPs. We've been producing RVPs of numerous viruses for 11 years, and this put us in a great position to rapidly respond to the SARS-CoV-2 pandemic. As soon as the genetic sequence was published, our molecular biology team was able to plug and play, and we produced infectious Wuhan Hu1 RVPs within weeks. We shipped out hundreds of RVPs and ACE2 cells for free to scientists all over the world, starting in May 2020. As everyone, even non-scientists, know, the world has experienced waves of variants in the past 2 years. This chronological phylogenetic tree shows the increasing number of mutations that the virus is accumulating, and the frequency figure at the bottom shows which variants have predominated over time. The world is now facing mainly Delta and its sublineages, shown in shades of yellow, and the virus is still evolving. We've kept our fingers on the pulse and our eyes on the news, and once a variant appears at a prevalence that is worth taking note of, or the World Health Organization declares a mutant a variant of interest or concern, we produce, QC, and ship it out to our customers as fast as possible. We currently have 67 SARS-CoV-2 variants in our catalog, in both GFP and luciferase reporters, and we've produced SARS-1 and MERS-2. We have an alanine scan library of SARS mutant RVPs too, and we happily take custom requests. In addition, our catalog contains dengue, Zika, chikungunya, and VSV RVPs, and we're adding more viruses soon. Our RVP variants enable neutralization profiling for monoclonal antibodies or vaccine responses in patient or vaccinee sera. Our reference antibody, in the left-hand figure, neutralizes the D614G variant as well as alpha, beta, and gamma variants. We tested a mAb from a customer in the middle figure, and it poorly neutralized the beta variant. Likewise, sera from a collaborator neutralized the D614G variant well in the right figure, but not the other three variants. This profiling is essential for insights into vaccine responses and therapeutic antibody efficacy. Our extensive catalog of RVP variants can help accelerate your vaccine and therapeutic antibody development in accordance with the FDA COVID-19 guidance documents. Our RVPs can be critical reagents in the development and testing of your vaccines and therapeutics. The FDA issued these guidance documents in an urgent response to the pandemic, and they offer recommendations for the production and use of these critical reagents. They are regularly updated as more variants appear, and adherence early in the development of your treatment can speed up progress in your applications. The FDA recommendations for vaccine developers state that vaccine developers should assess immunogenicity of modified vaccines against targeted variants. Neutralization assays with wild-type SARS-CoV-2 or pseudoviruses may be used, and immunogenicity studies should be sufficiently powered to demonstrate immunogenic non-inferiority to the original vaccine. For developers of therapeutic antibodies, the FDA recommends that sponsors should routinely monitor genomic databases for emerging SARS-CoV-2 variants, report the neutralizing activity of their antibody against circulating variants, and implement a potency assay to ensure that each lot is consistently produced and that the potency is maintained over the shelf life of the product. Our RVPs will enable you to fulfill these criteria, and links to these FDA documents are available on our website too. RVPs are not our only technology to support antibody development informed by FDA guidance. Our membrane proteome array can help address the FDA recommendation that preclinical studies should evaluate potential off-target binding of antibodies. Our membrane proteome array screens antibodies for off-target binding against 6,000 human membrane proteins. You will get an IND-enabling report in approximately 4 weeks, and in many cases, this report is accepted by the FDA in place of tissue cross-reactivity studies. Another recommendation is that antibody developers should thoroughly characterize epitopes to identify escape mutations in circulating variants. Our shotgun mutagenesis epitope mapping is a technology that rapidly maps antibody epitopes at single amino acid resolution. For SARS-CoV-2 antibodies, results are available within 4 weeks. Here at Integral, we are very proud of how our technologies have enabled scientists around the world to progress in the fight against SARS-CoV-2, and we look forward to working closely with you to ensure you succeed. Thank you.
Good morning, everyone. My name is Pavel Nikitin. I am a Senior Director of Antibody Engineering at Immunome, and today I would like to tell you how we are harnessing the human memory B-cell response to develop antibody-based therapeutics in our company. The next slide is disclaimers and forward-looking statements from our legal. Immunome is a vibrant public biotech company located in Philadelphia suburb. At Immunome, we study the human immune response to identify meaningful therapeutic targets and patient-derived antibodies against those targets. We study the human memory B-cell compartment in an unbiased manner to understand the diversity of the immune response to diseases, whether chronic or acute. So our platform allows us to see disease through the lens of the human memory B-cell. Our discovery process, shown on the right, starts with the isolation and immortalization of patient memory B cells from appropriate compartments that allows us to capture the immunoglobulin response to the disease. Immunome uses its highly efficient, high-throughput engine to isolate disease-specific antibodies, identify the antigens of those antibodies, and then validate these antibody-target pairs for potential therapeutic utility. It is important to emphasize that the output of our discovery engine includes both unique antibodies and their targets. And today's talk is focused on Immunome's COVID program and on the 3-antibody cocktail that is a part of our infectious pipeline. Immunome was awarded up to $17.6 million in funding from the Department of Defense to discover and develop anti-SARS-CoV-2 antibodies for the treatment of COVID-19. Throughout the talk today, I will highlight how we used integral moleculars technology to facilitate the development of our antibody cocktail. As you are well aware, the current pandemic remains a serious medical issue in the US and across the globe. While vaccines represent a critical tool to fight the virus, recent data demonstrate that emergent variants can evade the immune response invoked by the current vaccines. Here is the data on neutralization titers in vaccinated patients from one of the recent reports. As you can see from the shown graphs, responses to viral variants such as Beta and Delta were limited among individuals immunized with the vaccine based on the original variant of virus. We therefore believe that COVID-19 therapeutics, with the ability to treat emergent variants, will continue to remain a critical component of the response to the infection. Multiple variants emerged since the initial viral outbreak. As you know, the Delta variant sublineage, first discovered in April of this year, now accounts for over 99% of cases in the United States. And this really demonstrates how quickly new variants selected in patient population can efficiently spread and evade the immune response even in the vaccinated population. Therapeutics will be particularly important for high-risk patients who do not derive benefits from vaccines, for example, immunocompromised individuals. When we started our project, we thought of potential viral drift and focused on antibodies that would retain activity against potentially emerging variants. As a result, we developed the therapeutic antibody cocktail IMM-BCP-01 that has multiple virus suppressive properties. I will cover some of them in today's talk. We studied antibody neutralizing properties in the BSL-2 pseudovirus assay. For that, we used integral moleculars reporter virus particles, or RVPs, shown on the left. On the right, you can see the neutralizing titer of IMM-BCP-01 cocktail against the clinically relevant Delta variant pseudovirus. The marked gray area on the far right end of the graph shows the observed antibody exposure based on the 9 mg/kg dosing in hamsters. As you can see, the EC50 is 2.5 log lower the observed antibody exposure. Overall, we have tested 11 RVP variants from integral molecular and alongside several in-house made pseudovirus variants. IMM-BCP-01 fully neutralized all of those variants. You can find the complete data summarized in our manuscripted bioarchive. Recent studies, however, show that neutralizing properties are not sufficient in treatment setting in preclinical models. In fact, potent neutralizing antibodies with mutations in the Fc domain that silence the effector functions such as phagocytosis, ADCC, or complement activation were shown to lose efficacy in vivo when measured both for the ability to decrease viral load in lung of infected animals or to enhance overall survival in a mouse model. These data support the idea that effector functions positively influence overall efficacy of antibody. Knowing that, we evaluated the effect of our antibodies on the most relevant mechanisms of virus clearance. We observed a quite potent activation of classical complement pathway by IMM-BCP-01 cocktail in vitro. IMM-2190, the blue line, was the only antibody of the mixture that elicited a modest level of complement activation on its own. In contrast, 2 and 3 antibody cocktails induced a robust combinatorial enhancement of complement fixation. Similar results we obtained in phagocytosis and ADCC assays. While the response from individual antibodies varied depending on the assay, the 3 antibody mix, shown in the black line here, and here, demonstrated combinatorial enhancement of activity regardless of the measured effector response. Now, classical complement activation is driven by the C1 complex, and that complex recognizes 3-dimensional structure of Fc domains within the immune complex. Similarly, phagocytosis and ADCC are activated by the exposed Fc regions of the antibodies. So really all three immune responses depend on the conformation. We therefore next studied the conformation of the antibody spike complexes. And in order to do that, we went back to integral molecular and used their shotgun mutagenesis approach. On the left panel, you can see a typical workflow for the shotgun mutagenesis project. Briefly, each of our antibody of interest was screened against a library of cells expressing spike RPDs with a single point mutation. The clones that affected binding of our antibodies are summarized in the table shown on the right. The binding was compared to several positive control antibodies not shown here that helped to exclude point mutants with altered conformations. As you can see on the right, the selected antibodies bound to non-overlapping epitopes of the spike protein. And when we mapped the binding sites on the structure of the spike protein, we confirmed that the epitopes were in fact non-overlapping. Interestingly, 2253 antibody bound to a broadly conserved and cryptic epitope also present on spike of SARS-1. The binding of this antibody causes a non-ACE2 dependent neutralization via a novel mechanism involving spike conformational changes. 2190 antibody recognizes a composite epitope involving the receptor binding reach and an area adjacent to the receptor binding loop and competes with cellular ACE2 receptor. The third antibody, 2184, recognizes a broadly conserved epitope across COVID-2 variants. This antibody has the avidity-based binding mechanism. Thus, three antibodies have different mechanism of action. In fact, in additional studies shown in the manuscript available at bioRxiv, we demonstrated that IMM antibodies synergize to enhance neutralization of SARS-CoV-2 variants. The robust efficacy of our cocktail in vitro correlated really well with tremendous potency in vivo. Here we are presenting a comparison of published in vivo efficacy data on sotrovimab shown on the left panel with IMM-BCP01 data shown on the right. Sotrovimab is of course a virus GSK antibody being used under an emergency use authorization to treat COVID-19 patients. So, in prophylactic settings, sotrovimab led to a 2-log clearance at 5 mg/kg, the effect that pretty much was plateaued at this dose. Further dose increase to 30 mg/kg failed to improve viral clearance. In contrast, dosing with IMM-BCP01 led to a 2.2-log clearance at 3 mg/kg that was highly dose dependent. At 9 mg/kg, our cocktail cleared around 4 log of virus in lungs of infected animals. So these experiments used a reference virus or Washington isolate. We have also observed similar dose response against the beta variant in the same hamster model. Finally, we studied the selectivity of our antibody cocktail using integral molecular's membrane proteome array shown on the left. In brief, MPA has 6,000 human membrane proteins natively expressed in human cells. We observed that IMM-BCP01 antibody cocktail displayed a clean MPA profile, meaning that there was no binding to human proteins. In fact, the only identified protein that had a Fab-directed positive binding by the IMM-2190 and IMM-2184 antibodies was SARS-CoV-2 spike protein. Interestingly, IMM-2253 antibody was found to bind both SARS-1 and SARS-2 spike proteins, which is consistent with the results obtained via integral molecular's shotgun mutagenesis method. In summary, current COVID vaccines and antibody therapeutics are not sufficient to treat new variants of SARS-CoV-2 virus. Recent reports show breakthrough infection in vaccinated individuals. In addition, current therapeutics were developed based on virus neutralizing, not a holistic overview of virus suppressive mechanisms. In contrast, preclinical data on IMM-BCP01 shows potential to change standard of care. Particularly, the cocktail has 3 antibodies with multimodal action, including both a strong viral neutralization and viral clearance in vitro, and retains potency against former and current CDC variants of concern. Lastly, as it was publicly disclosed before, the IND submission for this project is anticipated in the last quarter of this year. With that, I would like to thank the organizers for the opportunity to present our story and you for your attention. I'll be happy to answer all questions during the upcoming Q&A session.
Hi everyone, my name is Yufei and I work in Dr. Ishii's lab in the University of Pittsburgh School of Medicine. Our lab is interested in developing mass spectrometry-based biotechnologies, and Antibody therapeutics. It's my pleasure today to share our work on the development of Nanobody pipeline technology and its therapeutic application on COVID-19. The pandemic has lasted for nearly 2 years, with more than 200 million cases worldwide. Though the COVID vaccines are available now and the trend in cases seems to be going down recently, there is still an urgent need to develop therapeutics that could potentially prevent disease progression or human-human transmission. Aside from the plasma or monoclonal antibodies isolated from the convalescent patient for treatment, an alternative solution is nanobodies. And for those who are not familiar with nanobody, nanobodies are the warheads of the heavy chain-only antibodies that are specific to the camelid family, such as llama, alpaca, and camel. Unlike IgG antibodies, nanobodies are small, only around 12 to 15 kilodalton. They are highly soluble and stable, and can be readily bioengineered into bi- or multivalent forms, and are amenable to low-cost, efficient microbial production. Due to their robust physicochemical properties, affinity-mature nanobodies can be applied by inhalation, making their use against the respiratory viruses very appealing. And these properties are also very important for drug delivery in vivo, storage, and transportation. For these reasons, nanobodies have emerged as a promising class of antibodies for biomedical applications and drug development. Inspired by Dr. Brian Chai's work, we have recently developed an advanced proteomics pipeline for nanobody-based drug discovery. In our pipeline, we immunize a domestic camelid to raise antigen-specific nanobodies by in vivo affinity maturation, and identify the nanobody sequences using the bottom-up proteomics approach. It may sound pretty straightforward, but there are several challenges for reliable identification and characterization of antigen-specific drug-quality nanobodies, including but not limited to the diversity and dynamic range of circulating antibodies in the serum, and also the unprecedented size and architecture of nanobody sequence databases. We successfully built a robust and highly reliable analytic pipeline and software algorithm to solve these problems and enable high-quality nanobody identification. I won't go into details here, but if you're interested, you're welcome to read our paper in the Cell System. The sensitivity and robustness of this approach were validated using antigens that span 3 orders of magnitude in immune responses, including a small, weakly immunogenic antigen. And the scale of the identification is also unprecedented. A surprisingly large cohort of single-chain nanobodies, typically thousands of distinct CDR3 nanobody families, can be identified. And the diversity of our identification is generally orders of magnitude higher than traditional methods such as phage display. In addition, we developed a strategy to quantify the repertoire of antigen-specific nanobodies. Tens of thousands of nanobody families were reliably quantified according to their desired properties, such as binding affinities. A significant fraction of the identified nanobodies has sub-nanomolar affinities for antigen binding, which are unusual for monomeric single-domain antibody fragments. We have also gained a plethora of interesting knowledges of mammalian humoral immunity. Our technology reveals at an unprecedented scale the efficiency, specificity, diversity, and versatility of antigen-engaged nanobodies that collectively shaped the comprehensive landscape of camelid antibody immunity. This provides unlimited possibilities for medical applications and drug discoveries. Last year, we applied our technology to the receptor binding domain of SARS-CoV-2 spike, and it was not surprising that we were able to identify more than 10,000 unique nanobodies with hundreds of highly diversified CDR3 clonal types. However, our lab had zero virology experience, and it was really challenging for us to characterize the neutralization activities of the binders. Luckily, I found a webinar of the integral molecular through the YouTube where I searched for the pseudovirus particles in the BSL-2 settings. The assay turned out to be extremely easy to follow with the step-by-step protocol as long as you have the experience with the cell culture. And most importantly, after confirming the infectivity, linearity of the pseudovirus particles, we were pleased to find out that the pseudovirus assay on my hands actually correlated pretty nicely to the conventional PRNT assay performed by our BSL-3 collaborators. And that's why we decided to stick to the company. By using the pseudovirus particles, we were able to verify dozens of potent SARS-CoV-2 neutralizing nanobodies on multiple epitopes of RBD, with the most potent one efficiently neutralizing authentic SARS-CoV-2 in vitro below 0.1 nanomolar. This most potent neutralizing monomeric nanobody we discovered also has outstanding physicochemical properties. It has extremely high affinity towards the RBD and also good thermostability and solubility. So we further engineered it to be a homotrimeric construct, which proved to have the best neutralization potency so far, and this construct remains ultra-potent even after aerosolization and lyophilization, making it a perfect drug candidate. We then explored the in vivo efficacy of our most potent multivalent SARS-CoV-2 nanobody. We call it inhalable nanobody 21, PIN21. The excellent physicochemical properties and small size of nanobodies raised an exciting possibility of efficient pulmonary delivery by aerosolization, with characteristics of rapid onset of action, high local drug concentration, and improved patient compliance that may benefit a large population of SARS-CoV-2 infected patients. In this study, we systematically evaluated the efficacy of our nanobody for treatment of SARS-CoV-2 infected hamsters, which model moderate to severe COVID-19 disease. By treating hamsters with our nanobody at an ultra-low dose, we observed a complete protection in the lungs of the animals with reversed weight change. Notably, there was a substantial protection of the upper respiratory tract of the animal as well. This is in sharp contrast with clinical monoclonal antibodies, which has no protection at all in the upper airways. Meanwhile, the structural analysis of the neutralizing SARS-CoV-2 nanobodies of different epitopes has revealed a novel conserved neutralizing epitope on receptor binding domain. These novel classes of nanobodies can fight against the emerging virus variants, and indeed, the virus of concerns are less resistant to nanobodies comparing to the IgGs isolated from the convalescent patients. And in summary, I hope my talk today has convinced you that our lab has developed a disruptive pipeline technology for drug quality nanobody discovery. By applying our technology, we're able to identify more than 8,000 high-affinity COVID-19 nanobodies against a receptor binding domain, and verified a panel of potent multi-epitope antiviral nanobodies. Our structural studies also reveal a novel class neutralizing nanobodies that bind to conserved epitopes and are resistant to the circulating variants. And most importantly, we have demonstrated the in vivo efficacy of our highly effective nanobody drug PIN21 through the novel inhalation treatment, which resulted in protection of both upper and lower airways with more than 6 log viral reductions. And I would like to thank my PI, Dr. Yi Shi, and also our lab members for the support of this work, and also our collaborators on the BSL-3 experiments, in vivo studies, and structure characterization. Thank you.
Thanks, Yufei, Pavel, and Haley for some really wonderful presentations. This is some seriously interesting work and makes me very, very optimistic about the future of COVID therapeutics. So thank you all very much for sharing that with us. To the audience, the Q&A is going to begin in just a few moments. So if you have a question for our panelists, don't hesitate. Send that in now. All you need to do is type your question into the Ask a Question box and then hit submit. Okay, so it looks like we have a bunch of questions for our panelists, so let's get to it and try to get through as many questions as we can. Bear with us for just a moment as we make the transition into the Q&A session. All right, everyone, thanks for joining us for the Q&A session. All of our panelists are here to answer your questions, try to get through as many of them as possible. But first, we do have a statement from Haley who'd like to address the emerging Omicron variant. Haley?
Thanks, Jeff. Thanks to everyone for joining us today. Before we proceed with the Q&A, I would like to give an update on the recently discovered Omicron SARS-CoV-2 variant. This variant has been designated a variant of concern by the World Health Organization. It has 32 mutations in the spike protein, and it has been detected in 14 countries around the world in the past week. Here at Integral, we are already in the process of making Omicron RVPs, and if you would like more information on them and their availability, please email us at info@integralmolecular.com. Thanks, Jake.
All righty, thank you very much for that very important information. We appreciate that. Haley, I'm going to go back to you. First question is going to be for you. One of our audience members would like to know, or they say that you mentioned that you take requests for custom RVPs. What's the timeline to generate a custom RVP?
Yes, we do take custom requests. You tell us what mutations you would like in your envelope protein, and we will design the plasmid. It takes about 6 to 8 weeks from designing the plasmid to shipping to you. And we're also available to schedule a Zoom call with you to talk in more detail about your needs.
All righty, thank you very much for that. Pavel, we have a question for you. An audience member says, have you used any of your variant RVPs for your studies, and what was your experience with them?
Sure. Well, first of all, I would like to thank organizers for inviting me and letting me to present our story. Yes, we certainly used the RVPs from Integral Molecular. I believe they showed some data on the Delta variant. Besides that, we used at least 10 more different RVP variants from Integral Molecular, and so far we have actually a very good experience. Similarly to the last speaker, we compared, I didn't show this data, but we actually compared the pseudovirus neutralization data and live virus neutralization for several strains, and we had a pretty good correlation between RVPs, pseudovirus neutralization, and the live virus.
Okay, thank you for that, Pavel. Uffe, over to you. An audience member says, what is the benefit of nanobody versus antibody therapy for patients? Would the inhalation delivery method have been successful with antibodies?
Well, so first I would like to thank for the opportunity of today's presentation. And there are several considerations when it comes to the therapeutics, the efficacy, the production, transportation, and storage. So when it comes to nanobodies, as I have mentioned in the talk, nanobodies, they're highly stable, and which makes the transportation and storage much easier. And they can be easily bioengineered into bispecific or multispecific, and they can be purified from microbes to drastically reduce the cost and effort for the production. And nanobody also possess similar good efficacy compared to antibody when we demonstrated the treatment efficacy in the animals. And when it comes to the inhalation delivery, you can imagine that like nanobody is only 1/10 of the size of the antibody. So it means that the higher concentration in a certain volume can be delivered for inhalation treatment. And in our experience, what we have observed was that when the proteins are in the same delivery buffer, the size of the protein is reversely correlated with the recovery from the aerosolization. It means that the larger the protein is, the more difficult it is to spray them for the aerosolization delivery. So being small and stable that could better resist the shear pressure from the nebulizer also makes Nanobody highly suitable for particularly the inhalation delivery therapy that can protect better the upper respiratory tract, at least in the animal studies. Yeah.
All right, thank you very much, Uwe. Haley, back to you. An audience member says, "Is there a titer for the strains different? Is the titer for the strains different than the wild type? And how do I know how much to order?"
Yes, the titer for the strains varies between the different strains and very slightly between our lots. Some of our RVPs, some of the variants are more infectious than the Wuhan strain, and some are less infectious. But every single lot in our catalog comes with a tech data sheet that is on our website. And the tech data sheet lists the recommended input per well for a 96 well plate. So that's how you can calculate how many mils of our RVPs you will need to order. And all this information is on our website.
All right, thank you. Pavel, over to you. What are the limitations of producing pseudovirus particles in-house?
So I would say it's just two different approaches that we took. We definitely produced some pseudoviruses in-house, but what we and why we actually used integral molecular was the speed and reliability. As you can imagine, making 10 to 11 different strains plus you need to constantly make a new strain once it appears is a lot of work. And from our perspective, it was easier to outsource this work to integral molecular. And so far, we are very happy about the quality of reagents provided.
All right, thank you, Pavel. Uwe, question, a good question, something actually I had in mind to ask. Audience member would like to know, are you planning to develop a nanobody cocktail?
I think the reason for developing cocktail is basically to block the mutational escape. In our case, nowadays they're easily to be engineered to be bispecific and cover different epitopes. So I didn't show data today, but we have data to show that our bispecific nanobody can actually block the mutational escape and be highly effective in animals. So yeah.
Okay,
but
thank
cocktail,
you.
yes. Yeah, maybe that's another possibility in the future. Yeah, but now a single nanobody, engineer form nanobody would be sufficient.
All right, thank you for that. Haley, question to you. Do you currently offer RVPs for other viruses?
Yes, we have RVPs for dengue, for all 4 serotypes of dengue. We have RVPs of Zika and of chickpea, and we are currently developing new RVPs for as yet unannounced viruses. And our entire catalog is on our website, and we also send regular emails out. And so you can get on the list if you'd like to hear about the new RVPs we're making.
All right, thank you for that. Our next question is for Pavel. Pavel, I would like to know, does the FDA have a preference to use live versus pseudotyped virus for neutralization studies?
Right. So there are several guidelines published. The short answer is FDA prefers to have both pseudovirus and live virus neutralization studies, but if a company has one or another, FDA also takes it. Again, there are published guidelines that one can refer to.
Okay, thank you. Uwe, how do RVP assays compare with PRNT assays for variants?
We have just finished a PRNT assay of 17 nanobodies neutralization against the Delta variant. And as far as the data shows that the correlation is still good, at least for the Delta. And for the other variant, for the pseudovirus, we have performed against beta, alpha, lambda, and all these variants, we see a strong correlation of their binding and neutralization, which means that if the nanobody can bind well to the RBD with the particular variant, they can show good neutralization in the pseudovirus. And if we see a decreased or abolished binding, then we can see attenuated, decreased neutralization affinity in the pseudovirus. So I would say they generally correlate pretty nicely.
Okay, thank you. Haley, we have a bunch of questions for you. First one here, an audience member says, does the, I'm sorry, I think we already asked that one. Sorry about that. Next question we have for you is, do you have already any neutralization profile data concerning the Omicron variant? Which I think you may have addressed, but I don't know if you have anything more to say about that.
So we don't have any neutralization data in-house yet. The sequence was only released to the world last Thursday. So we have started producing the DNA to make these RVPs. So we don't have neutralization profile data, but we expect to in the next few weeks.
And then as a follow-up to that, Haley, we have another question. An audience member says, are there any news about different properties of the Omicron variant? So vulnerability to the new version of, or vulnerability of the new version of the SARS-CoV-2 virus?
Well, this is such a new variant that has got the whole world interested and worried. So the data is really limited, but what we do know already is that Omicron has 32 mutations in the spike protein, and that is more than double the number of mutations in the spike protein of Delta. It has a really interesting combination of mutations. It's got mutations that are present in a lot of other concerning variants. For example, it's got N501Y, which is also seen in alpha, beta, and gamma. It's got mutations at E484, which is also seen in beta. However, we don't quite know what the combination of all these mutations does. Because the majority of new sequences coming out of South Africa, for example, are Omicron over Delta, there are suggestions it's more transmissible. So these mutations may increase transmissibility, and it may be more transmissible than Delta because it's outperforming Delta. We don't know if Omicron evades vaccines, and that's obviously very concerning because the population of South Africa doesn't have a very high percentage of people vaccinated. I think it's probably 25 to 35% of people. So we don't know if it evades vaccines. It's spreading in a country where there's not a lot of people are protected. The whole world is trying to figure things out right now. There have been some very early studies suggesting that your chance of reinfection is higher if you have Omicron. There are suggestions that the demographic of who the virus is infecting is changing. 20 to 30-year-olds are showing moderate to severe disease symptoms. We don't know about, as the previous question was, about the neutralizability yet, but what is interesting is that Delta has 2 mutations in the receptor binding domain, while Omicron has 10 mutations. So, yeah, we're figuring it out. I feel like every virologist is working on this right now, so we're getting answers so fast. But, yeah, that's what we know right now.
Thank you, Haley. Appreciate that. That's important information. Next question we have for you. An audience member says, can the RVPs be used to test cross-neutralization titers in convalescent sera from patients infected with different SARS-CoV-2 variants?
Yes, absolutely. And that's why our catalog is so useful, because we have so many different variants that you can test. So yeah, sera can be tested for neutralizing titers currently on the 67 strains we have available, and soon on the Omicron variant.
All right, thank you for that. Next question we have is, oops, if my screen will cooperate with me here, is a logistic question where an audience member says, what is the wait time between order and shipment?
So if you place an order with Layla at sales@integralmolecular.com by Friday at noon, we will ship your order on Monday or Tuesday the following week if we have your request in stock. If it's a custom, as I said earlier, it's 6 to 8 weeks. But yeah, if you get your purchase order to us by Friday, we will ship Monday or Tuesday. And we can also ship on other days of the week if you are international and it suits you to receive it on a different day.
Right, great. Looks like we have 2 more questions for you. Next question, audience member says, do you have customers that have used your RVPs in vaccine clinical trials?
Yes, we do. However, we don't have any data that we can publicly share right now with respect to SARS-CoV-2, but there is a customer of ours, Takeda, who have used our Zika RVPs in a phase 1 clinical trial successfully, and that was published in the Lancet Infectious Diseases journal earlier this year.
All right. And looks like the last question we have, which is actually a very important one as well, what will be the biosafety level for RVPs? Will they be the same as the original virus?
And perhaps I can take that question and put my virologist hat, since I got degree in virology. For RVP, the biosafety level is BSL-2, and that's how it is intended to use. That's how it's usually used. And I'd like to refer to Integral Molecular's presentation. These RVPs were made from the replication incompetent lentiviral-based particles. So they are safe to use in the BSL-2 environment.
All righty, thank you for that, guys. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on the GEN website at genenginews.com for up to a year. So if you missed any parts of it, you can watch it again, or feel free to forward the link to any of your friends and colleagues who you think might be interested in this presentation. I'd like to thank Haley, Pavel, and Uffe again for their really amazing and in-depth presentations and all the information, especially on the new Omicron variant. We appreciate that. And I'd like to thank you, the audience, for your attention and very thoughtful questions. Very special thanks to Integral Molecular for sponsoring this webinar. Hopefully we'll see you all again at another GEN webinar in the near future. Goodbye for now. Everyone stay safe and healthy and get your vaccines.